A very good evening to all. So today we are going to discuss some of the questions which came in test. So quickly answer, how was your test? Good evening all. So how was your test? Was it easy, moderate or difficult? Okay. So now let's discuss some of the question. Question number 1 says, select the incorrect match from the following. Option A is tulip, bisexual flower. Option B is petunia, endospermous seed. Option C is aloe vera, calyx 3+3 and corolla 3+3. And D is tobacco, epipetalus androecium. What do you think what will be the correct option? They're asking about the incorrect match. You know that petunia and tobacco, they both belong to family Solanaceae. Tulip and aloe, they belong to family Liliaceae. Okay. Tulip is a bisexual flower. Yes, correct. Petunia has endospermous seed. Yes. Tobacco, epipetalus androecium. Yes. Epipetalus means when the stamens, they are attached to the petals. Okay. We call this condition epipetalus. But aloe vera, remember Liliaceae, they have perianth. Where the calyx and corolla, they are not differentiated. And the unit of perianth is tepal. Okay. So in aloe vera, the tepal shows 3+3 arrangement. There's no differentiation of calyx and corolla. So this is incorrect. And a correct option with respect to question, because question is asking about the incorrect match. I hope it is clear. Moving to next. All of the following statements are true with respect to aleuroplast except, you know that plastids are of 3 types. There's chloroplast, there's chromoplast, and the leucoplast. Okay. So quickly answer what you think what will be the correct option for this question. Here option A is given that it is a double membrane bound structure. Option B says it contains protein digesting enzyme. Option C says it is found in seed. And option D says it is a colorless plastid. So what do you think what will be the correct option for this question? B, very good. Chloroplast, as I told you that plastids are of 3 types, the chloroplast, the chromoplast, and the leucoplast. Chloroplast is consists of chlorophyll plus carotenoids. Chromoplast is consists of carotene and xanthophyll. And leucoplast, they are colorless plastid. And leucoplast is further of 3 types. Remember this beta, they are very important. Number 1 is aleuroplast, which is given in the question, which stores protein. Next one is aleoplast, which stores oil. A myeloplast, third one, which stores starch or carbohydrate. Now let's come to option. Option says a double membrane bound structure. Yes, because it's a plastid. It contains a protein digesting enzyme. No. It stores protein. It does not contain the protein digesting enzyme. It is found in seed. Yes. It is a colorless plastid. Yes, because it's a part of leucoplast. A leucoplast is a colorless plastid. So all of the following statements are true with respect to aleuroplast except B, which says that it contains protein digesting enzyme. Now moving to question number 3. Question number 3 says during the post-transcriptional modification, splicing does not occur in cells. Option A says E. coli, B. plants, C. algae, and D. animals. Quickly answer. What do you think? What will be the correct option? They're asking about during the post-transcriptional modification. Splicing does not occur in. What do you think? What will be the correct option? No, beta, it's not C. See, there are two types of cells, prokaryotic and eukaryotic. Okay, the prokaryotes and the eukaryotes. We know that eukaryotes are more complex as compared to prokaryotes, right? E. coli is a prokaryote. It's a bacteria. And plants, algae, animals, they all are eukaryotes. Right? Prokaryotes, they don't show post-transcription modification. There's no post-transcriptional modification. Okay, if there's no post-transcription modification, that means there would be no splicing. And splicing is the removal of introns. As I told you that eukaryote are more complex. They have exon and they have introns, right? Exons and intron. Introns are the known coding sequence which does not code for any functional protein, and exons, they are the coding sequence which codes for a functional protein. The eukaryote, they have exon and introns. So splicing takes place in eukaryote. And splicing is the removal of this intron so that two exons can join and we can get a complete coding sequence. So splicing takes place in eukaryote. Capping takes place in eukaryote, which occur as 5' end. Tailing takes place in eukaryote, which occur at 3' end. Remember, in eukaryotes, this is a DNA. This DNA is transcribed into mRNA. This process is known as transcription. And this process is taking place in the nucleus. Whereas in prokaryote, this process is taking place in the cytoplasm. Okay, and translation is also taking place in cytoplasm in case of prokaryote. But in eukaryote, because it's a complex organism, they have their transcription takes place in nucleus and translation occurs in cytoplasm. So when this mRNA is formed, when it is in immature stage, we call it as hnRNA. Now this hnRNA undergo modification, or we call it as post-transcriptional modification. This hnRNA undergoes capping, tailing, splicing. And after these post-transcriptional modification, we get the mature mRNA, which is mRNA, which undergo translation. Okay, so post-transcription modification and splicing only taking place in eukaryotes. In prokaryote, there is no post-transcriptional modification and there is no splicing because they does not have introns. Okay, so the question is asking during post-transcriptional modification, splicing does not occur in cells. It does not occur in prokaryote, which is E. coli. I hope it is clear to everyone now. Moving next, Identify the figure given below and select the incorrect match of the label parts A, B, C, and D with their respective description. Okay, so this is a human excretory system is given, and they're asking you the A, B, C, D with their respective description. Option A is given, B is given, C is given, and D. Option A says A, urine produced by kidney exit through this and passes into the common sac. Option B says during urination, urine is expelled from the bladder through this. C says it secretes antidiuretic hormone when blood osmolarity rises above a set point. Option D says the structure that stores urine. What do you think? What will be the correct option for this question? Quickly answer. This I told you when I was discussing, when I taught you the human excretory system. Yes, very good. C represent adrenal gland. They are present on the anterior part of the kidney. A represent ureter. D represent urinary bladder. And B is urethra. Okay? Now, produced by kidney, exhibit through this and passes into a common sac. Yes, it's true. B says during urination, urine is expelled from the bladder through this. Yes, true. Option D says the structure that store urine. Yes, D is the structure that you store urine, which is urinary bladder. But option C says that it secretes antidiuretic hormone when blood osmolarity rises above a set point. Says that it secretes antidiuretic hormone, which is ADH. Another name of ADH is vasopressin. Beta, ADH and vasopressin, they are synthesized by hypothalamus. ADH, or we also call it as vasopressin, it's synthesized by hypothalamus and stored in the posterior pituitary. Remember this. So it gets stored in the posterior pituitary. When the posterior pituitary receives the signal from hypothalamus, posterior pituitary releases this vasopressin. And vasopressin role is in conditional reabsorption of water and electrolyte. So it's maintain the balance of water and electrolyte in the body fluid. That is the main function of ADH or vasopressin. But here it says that it secretes antidiuretic hormone and sees the adrenal gland. Adrenal gland does not secrete ADH hormone. Remember this. It is synthesized by hypothalamus, stored in posterior pituitary, and posterior pituitary releases this ADH. Okay. So question is asking about the incorrect match, which is option number C. I hope it is clear to everyone. Now moving to the next one. Seeds in which of the given plant has no residual endosperm as it is completely used up during embryo development? Option A is groundnut, B barley, C castor, D maize. What do you think what will be the correct option for this question? Try to recall the endospermous seed and non-endospermous seed. Then you can easily answer this question. What do you think what will be the correct option for this question? Okay. As you know, there are two types of seed. There are endospermous seed and there are non-endospermous seed. Okay. Endospermous seeds are those that have endosperm for nourishment, because endosperm provide the nourishment to the developing embryo. And non-endosperm, where the endosperm is completely utilized. So there's no endosperm. And the question is asking seed in which of the plant has no residual endosperm. That means they're asking about non-endospermous seed. Right? And generally, monocot seeds are endospermous seeds. And dicot seeds are non-endospermous seeds. Okay? But remember exception. Okay? In monocot seed, exception is orchid. Orchid is a monocot, but it has non-endospermous seed. And in dicot seed, the exception is castor. Castor is a dicot seed, but it has endosperm. Okay, now you can quickly answer. Barley is monocot. So monocot has endospermous seed, and question is asking about non-endospermous seed. Okay, so barley is not a correct option. Maize is also a monocot, and monocot seeds are endospermous seed. Castor is a dicot, but it's an exception because castor has a endospermous seed. It is not. So the rest option is A, which is groundnut. So the correct option is groundnut because groundnut has non-endospermous seed. Now moving next. A mixture containing DNA fragment P, Q, R and S with size of P plus R equals to Q, where P is greater than R, S is greater than Q, was subjected to agarose gel electrophoresis. The position of these fragments from cathode to anode side of the gel would be. So they're asking about the position of the fragments, DNA fragments. Okay. So what do you think? What will be the correct option or the correct arrangement or the sequence of the fragment? Okay. They are saying that there are 4 fragments. Okay. There's P, the Q, R, and S. Very good. And they're saying that the size of the P plus R equals to Q. And P is greater than R and S is greater than Q. This is what is given in the question. Okay. Now let's assume that P is 3. Okay. I'm hypothetically given the numerical value. Okay. Let's assume that P is 3. Wait. Let's assume because R is smaller, so let's keep R is 3, P is 5. Okay. Now let's assume S is greater. So let's keep that S is 10 and Q, which is given that P plus R equals to Q. So if I add 5 plus 3 equals to 8. So Q is 8. Okay. Hypothetically, I have assigned these values. This is a agarose gel. electrophoresis. Okay, here are the wells in which we are going to load the sample. In this test tube, there's a DNA which is treated with the restriction endonuclease. Okay, so this is a DNA fragment. When restriction endonuclease act on it, it will cut this DNA fragment into 4 fragments, which is P, Q, R, S. Okay, and they are asking the arrangement from the cathode to anode. So cathode is negative and anode is positive. And we know that the DNA fragment is negatively charged, so it will always move from negative towards positive. It will always move towards the positive because DNA is negatively charged because they have phosphate group. Okay. I loaded the DNA sample in the wells. Now these samples will start migrating. They will start moving from cathode to anode. So the fragment which is larger in size will travel less. Okay. And which one is larger in size? It's 10. It's S because it's having a value 10. So I write S. S will travel the less distance because it is larger in size. Then followed by Q. Least is R. So R will travel the maximum distance because R is having a very small size. And the fragment which are smaller in size, they will cover a larger distance as compared to those which are larger in size. So R will travel the maximum distance. So R will quickly reach the anode. Left is P. So the P will obtain a position between Q and R. So the sequence will be S, Q, P, R from cathode to anode. So the correct option will be B, Q, S, Q, P, R. I hope it is clear to everyone now. Moving next. Match the following column with respect to various scientists and their respective discoveries. In column 1, scientist name is given. In column 2, their discoveries are given. Okay, and you have to find out the correct option by matching the column 1 with column 2. So FW went, he worked on oxygen. Remember the agar block? What he did, first he exposed the colloidal tip with the light. So this colloidal tip starts synthesizing oxygen. So oxygen is present in the tip. Okay. Now what he did, he removed the tip and placed it in a medium which is containing agar. So all the oxygen which are present in the tip, they will start diffusing into the agar. Now he take the agar block and placed it on the decapitated stump. When he placed the agar block, which is already containing the oxygen, this oxygen will start transporting downward and get accumulated on the darker side. Later, what he observed is the curvature. Because the side where the oxygen transport and get accumulated, oxygen main function is the cell elongation, cell enlargement, and it promotes cell division. So all the cell in this region, they start dividing, they start increasing in length. So bending take place, or we obtain a curvature. This what FW went observed, and he worked on oxygen. So A3. Cousin, he discovered ethylene. He observed that from the ripened oranges, some volatile compound is coming out, and this volatile compound is responsible for the ripening of unripe bananas. So later he find out that this volatile compound is ethylene. E. Kurosawa, gibberellic acid, C1. He observed the Bakane disease, the foolish rice seedling, in foolish seedling disease in case of rice. Now, F scoop, he discovered chitin. So A3, B4, C1 and D2. So correct option would be C. I hope it is clear to everyone. Moving next. Which among the following bacteria carried out denitrification? A. Nitrosomonas, B. Nitrococcus, C. Thylobacillus, and D. Nitrobacter. What do you think? What will be the correct option for this question? They're asking about denitrification. Quickly answer. Very good. As you know, that first is ammonia is oxidized to nitrite, right? Nitrite is oxidized to nitrate. In this process and this process and this process, both are the part of nitrification. When ammonia is converted to nitrite, in this process, the bacteria involved are Nitrosomonas and Nitrococcus. And when nitrite is converted to nitrate, here the bacteria which is involved is Nitrobacter. And this nitrate is absorbed by plants. But the nitrate which is present in the soil is reduced to nitrogen. And this process is known as denitrification. This process is carried out by two bacteria. One is Pseudomonas and another one is Thiobacillus. Okay, so question is asking which among the following bacteria carried out denitrification is option number C, which is Thiobacillus. Okay, I hope it is clear to everyone. Moving next, consider the given statement carefully. 1. Malphizian tubules help in the removal of nitrogenous waste and osmoregulation in periplanety. Option 2 says, statement 2 says that flame cells are the excretory structure in planaria as well as rotifer. Statement 3 says that each kidney of an adult human measures 0.1 to 0.12 meter in width. Statement 4 says the kidney are situated between the level of the last thorax and the fourth lumbar vertebrae. Select the option with correct statement. They're asking about the correct statement. Quickly answer what you think what will be the correct option for this question. Very good. See, the question is saying that Malphizian tubules help in the removal of nitrogenous waste and the osmoregulation in periplanety. Malphizian tubules, they are present in cockroach, right? And periplanety, they are is a genus of cockroach. American cockroach. So statement 1 is correct. 2 says the flame cells are the excretory structure in the planaria as well as rotifer. Yes, it is true. Third says that each kidney of an adult human measures 0.1 to 0.1 meter in width. No, it is 0.5 to 5 to 7 centimeter. Centimeter in width. Kidney are situated between the levels of last thorax and the fourth lumbar. No, it's a third lumbar. So 1 is correct and 2nd is correct. So correct option is B because they're asking the correct statement and B is that statement 1 and 2 are correct. 3 and 4 are wrong. So the correct option would be B. I hope it is clear to everyone now. Now moving to next one. Read the following statement and select the correct option. Statement A says the mitosis is seen in both diploid and haploid cells. Statement B says the meiosis involves 2 sequential cycles of nuclear and cell division, but only a single cycle of DNA replication. Option A says that only statement A is correct. Option B says only statement B is correct. Option C says both statements are incorrect. And option D says both statements are correct. What do you think? What will be the correct option for this question? They're asking about the mitosis and meiosis. So in mitosis is seen in both diploid and haploid cells. Yes, in plants you can see the mitosis is occurring in both diploid and the haploid cells. Meiosis involves 2 sequential cycles of nuclear division and cell division. Yes, there's meiosis 1. Remember, there's meiosis 1 and there's meiosis 2. But only a single cycle of DNA replication. Yes, only a single cycle of DNA replication. So both the statements are true. These are the direct statement from NCERT. Okay. So statement A is true, statement B is true. So option would be C, that both so option would be D, that both statements are correct. Okay, I hope it is clear to everyone. Now moving next. Colloid roots A are found in pinus, B have fungal association, C are associated with nitrogen fixing cyanobacteria, D possesses root hair. Quickly answer what you think what will be the correct option for this question. They're asking about the colloid root. Try to recall gymnosperm. Colloid roots, these are found in cycas. Okay? And these colloid roots are formed with, they are associated with the nitrogen fixing cyanobacteria. So they show association with nitrogen fixing cyanobacteria. Remember this, that colloid roots are found in cycas. In case of pinus root, they show association with fungi and in the form of mycorrhiza. This association is known as mycorrhiza. Okay, so the colloid roots, option C is the correct answer, are associated with nitrogen fixing cyanobacteria. Moving next, select the odd one out with respect to life cycle pattern of the given algae. Ectocarpus, Polysiphonia, Spirogyra, and Kipps. What do you think what will be the correct option for this question? They're asking the odd one out with respect to life cycle pattern. No, it's not D. See, the ectocarpus, Polysiphonia, calp, all these, they have haplodiplontic life cycle. Okay? Remember, fucus, the brown algae, it has diplontic life cycle. But Spirogyra, which is a green algae, it has haplontic cycle. Mostly all algae, they have haplontic life cycle, but the exceptions are given in NCERT that Ectocarpus, Polysiphonia, kelps, they all show haplodiplontic life cycle. Fucus is also an algae, it shows diplontic life cycle. Please remember these exceptions, these are very important. So select the odd one out with respect to life cycle pattern of the given algae. So option is 3, which is Spirogyra, because Spirogyra have haplontic life cycle, whereas Ectocarpus, Polysiphonia, kelps, they have haplodiplontic life cycle. So odd one out is Spirogyra. Now moving to next. The end product of glycolysis. Option A says enters mitochondrial matrix and undergo oxidative decarboxylation. Option B says is a 2-carbon compound. Option C says forms 3-carbon acetyl-CoA. Option D says enter directly Krebs cycle. What do you think? What will be the correct option for this question? No, it's not C. The end product of glycolysis. In glycolysis, glucose is broken down into pyruvate. And pyruvate is a 3-carbon compound, right? This pyruvate will move into mitochondria. In the matrix of mitochondria, the pyruvate will be converted to acetyl-CoA. This acetyl-CoA will enter into the Krebs cycle. Okay, so when pyruvate is converted into acetyl-CoA, acetyl-CoA is a 2-carbon compound, and 1 carbon is released in the form of carbon dioxide. The enzyme which catalyzes this reaction is known as pyruvate dehydrogenase. Okay. Option 1 says the enter mitochondrial matrix. Yes, pyruvate. The end product is pyruvate. It entering into the mitochondrial matrix. Undergo oxidative decarboxylation. Yes, it undergoing oxidative decarboxylation, where acetyl-CoA is formed and decarboxylation is written. That means CO2 is going to evolve or released. It is a 2-carbon compound. The end product of glycolysis is not a 2-carbon compound. It's a 3-carbon compound, which is pyruvate. It is wrong. Forms 3-carbon acetyl-CoA. No, it forms 2-carbon acetyl-CoA. This is wrong. Enter directly Krebs cycle. No. First, pyruvate is converted into acetyl-CoA. Acetyl-CoA will enter into the Krebs cycle. So the correct option is A. It enters the mitochondrial matrix and undergo oxidative decarboxylation. I hope it is clear to everyone. Now moving next. Match the column and select the correct option. Mycoplasma. B is nostoc, C euglena, D slime molds. In column 2, it is given spore-producing eukaryote, wall-less prokaryotes, oxygenic photoautotrophs. Fourth is wall-less autotroph. What do you think? What will be the correct option for this question? Okay, first is A, mycoplasma. So mycoplasma is also known as PPLO, which is pleuropneumonia-like organism, which is also known as joker of the cell. This mycoplasma is pathogenic, both to plants and animals. Mycoplasma lacks cell wall. They does not have the cell wall, and it's a prokaryote, right? So mycoplasma, wall-less prokaryote, A second. Nostoc. Nostoc comes under cyanobacteria, right? And Nostoc, they are photogenic they perform photosynthesis. So they are oxygenic photoautotrophs. B3. Luglina. Luglina is also photosynthetic in nature, and it does not have a cell wall. It has a protein layer which is known as pellicle. Remember this. So they are wall-less autotroph. C fourth. Slime molds, they are spore-producing eukaryote. So D1. A second, B third, C fourth, and D1. So the correct option is B. I hope it is clear to everyone. Now moving next, match the following column and select the correct option. Column 1 containing lysosome, inclusion body, smooth endoplasmic reticulum, and D vacuole. In column 2, it is given synthesized steroidal hormone contain excretory product formed by the process of packaging in Golgi apparatus, not bound by any unit membrane. Now, what do you think what will be the correct option for this question? So we know that the drawing nucleus, the nucleus, and the outer membrane of the nucleus is continuous with the ER. It's a ER. This is a Golgi. This is a cis phase of Golgi, and this is a trans phase of Golgi. So ribosomes which are present on the ER, we call this ER as rough endoplasmic reticulum. So it will synthesize protein. This protein, so vesicle will bud out from the endoplasmic reticulum containing the protein. This will be received by the cis phase of the Golgi apparatus. And the Golgi main function is the modification of the protein or lipids which are received from the endoplasmic reticulum. And from the trans phase, the vesicle will come out which contains all the modified proteins. Okay, this vesicle contains the modified protein. Or it could be a vesicle which contains the modified lipid. So if this vesicle contains the hydrolytic enzymes, now we call this vesicle as lysosome. Okay. So lysosome is formed by the process of packaging in the Golgi apparatus. So A would be 3. Inclusion body, they are not bound by minute membrane because these inclusion body, they are found in prokaryotes. They are not bound by any unit membrane. They lie freely in the cytoplasm. For example, like cyanophycine granules. Okay. So inclusion body B is not bound by any unit membrane. B fourth. Smooth endoplasmic reticulum. Main function of smooth endoplasmic reticulum is the synthesis of lipids, synthesis of steroid hormone, and detoxification of drugs. So C is 1, which is synthesized steroidal hormone. D, vacuole. Vacuole contain excretory products. So it's C2. A3, B fourth, C1, and D2. So the correct option is B. I hope it is clear to everyone. Moving next is question number 16, which says the relationship between the species richness and area represented in the following graph is described by which of the given mathematical expression? Okay, so this graph shows you a relationship between the species richness and the area. Now let's see. This graph, we know that when the area increases, the species richness also increases. So this graph is a rectangular hyperbola and it represents the equation. Let me draw it here. This is species richness, this is the area. Okay. So this represents S equals to CAZ, where S represents the species richness and A is the area. But if I draw this relationship on the log scale, I obtain this straight line, and which is represented by log S equals to log C plus Z log A, where S is the species richness, C is the y-intercept, Z is the slope of line or the regression coefficient, and A is the area. So the correct option is option number B, where S equals to CAZ, because it's a rectangular hyperbola. If the straight line is given, that means they're representing the relationship between the species richness and the area. On the log scale, this relationship would give you a straight line. Then the correct answer would be A. But they have given you this rectangular hyperbola, so it will be B. Okay. Now moving next. In the structure of DNA, N-glycosidic bond joins A, two nitrogenous bases, B, purines to ribose sugar, C, pyrimidine to pentose sugar, D, nucleoside to phosphate. What do you think what will be the correct option for this question? In the structure of DNA, N-glycosidic bond, remember, nucleoside is when sugar plus nitrogenous base. We call it as nucleoside. And nucleotide is when you have sugar plus nitrogenous base plus phosphate group. Then we call it as nucleotide. Okay, remember this. And between nitrogenous base and sugar, the bond is glycosidic. The bond between the two nitrogenous bases is hydrogen bonding. Between the sugar and the phosphate is phosphoester bond. Okay, remember this. In the structure of DNA, they're asking about the DNA, N-glycosidic bond join, N-glycosidic bond. That means they're asking the bond between the sugar and base. Two nitrogenous bases? No, there's a hydrogen bonding between two nitrogenous bases. Purines to ribose sugar. In DNA, there's a ribose sugar is there, but it is deoxyribose. Remember this. In DNA, it is deoxyribose. In case of RNA, it is a ribose sugar. But they're asking about the DNA. So ribose sugar could not be a correct option. Option C says pyrimidine to pentose sugar. Yes. Pyrimidine is nitrogenous basis and to pentose sugar. Deoxyribose is also a pentose sugar. And this is also a pentose sugar. Nucleoside to phosphate, no. So the correct option is option number C. Moving next, select the incorrect statement with respect to transpiration. It is the evaporated loss of water by plants. It transports mineral from the soil to all parts of the plant. It keeps the cell flaccid. It cools the leaf surface. Select the incorrect statement. They're asking about the incorrect statement. So what do you think what will be the correct option for this question? Try to recall the transport chapter. Role of transpiration. Functions of transpiration. Now you can easily answer. It is the evaporated loss of water by plant. Yes. It transports mineral from soil to all the parts of the plant. Yes. It keeps the cell flaccid. No. It keeps the cell turgid. Okay. This is a plant. If the continuous evaporation is taking place from the surface of the leaf, it creates a suction pressure in the vessel, because of which the plant roots, they absorb maximum water. Now the each cell is getting a water. That means the cell would be turgid in nature. Okay, so it keeps the cell turgid. It cools the leaf surface. Yes, it keeps the leaf surface cool by evaporative cooling. So select the incorrect statement. Incorrect statement is C. It keeps the cell flaccid. Question 19 says, match the column 1 with column 2. Select the correct option. They're asking about the correct option with respect to stomach. A is mucous neck cell, B oxantic cell, C chief cell. In column 2 is given mucus, intrinsic factor, pepsinogen, and HCl. So in stomach, the gastric rugae are present, right? The gastric rugae, they consist of gastric glands, and they have 4 types of cells. There's mucous neck cells, which will produce mucus. There's oxantic cell, or we also call it as parietal cell. They will produce HCl plus intrinsic factor, which absorb vitamin B12. Third one is peptic cell. We also called it as chief cells. They will produce pepsinogen. Fourth is G cell. The G cell produces a hormone which is gastrin. And gastrin stimulate the secretion of gastric juice. Now let's match the option. A says mucous neck cell. A, mucous neck cell will secrete mucus, so A1. Oxantic cell, they will produce HCl and intrinsic factor, so second and fourth. Chief cell will produce pepsinogen. Okay, now match. Here A is 1, B 4th, but C is 2nd, so this is not right. A1, B2, C3, so the correct option would be B. Okay, I hope it is clear to everyone. Moving next, an injury or a trauma stimulate dash in the blood to release dash which activate the mechanism of coagulation. Choose the correct option that fill in the blanks respectively. The injury or the trauma, they stimulate platelets or thrombocytes in the blood to release thromboplastin. Let me draw a flow chart for you. If there's an injury will activate or stimulate the thrombocytes. Thrombocytes are also known as platelets. These thrombocytes will release thromboplastin. This thromboplastin will attract more platelets, and these platelets will now release more thromboplastin. Okay. Now this thromboplastin will activate thrombokinase. Thrombokinase will act on prothrombin. Prothrombin will be converted to thrombin. Now this thrombin will act on fibrinogen and convert fibrinogen to fibrin, and fibrin result in a blood clotting. I hope it is clear now. So the correct option would be B, thrombocytes, and they will release thromboplastin, which activate the mechanism of coagulation. I hope it is clear now. So already 6, we are winding up the class. Thank you all for your participation. Thank you all and see you in the next class.